The immune system and leukocyte function. Each major component of the immune system is critical for human survival. The immune activity protects against infections that would be lethal without immune defense and eliminates cells in the stages of cancerous transformation. Primary immunodeficiency disorders, which often present in childhood, are a result of genetic mutations. Even though they are rare, they can be very severe. The most common immunodeficiency disorder occurs secondary to HIV infection. Pathogens express virulence factors that allow them to outcompete their counterparts, and in doing so, cause damage and disease. To defend against pathogenic microbacterial attack, as well as maintain homeostasis with other organisms, human beings have evolved a highly complex and orchestrated system of cells, tissues, and organs that are collectively known as the immune system. Cytokines are circulating protein molecules that are produced and secreted by immune cells to promote the proliferation and activation of other immune cells. The cells of the immune system are equipped with cell surface receptors and secreted molecules that act as sensors to detect and respond to foreign invaders or non-self molecules and to molecules that are commonly associated with pathogens. The immune system is capable of responding to abnormal self molecules, such as any mutated self proteins that are expressed by tumor cells, or organs that are received from a donor when transplantation occurs. The cells of the immune system circulate in the bloodstream to the body tissues and through the lymphatic system back to the bloodstream, conducting continual surveillance and attacking any foreign invaders. Failure of the immune system to function properly can result in aberrant immune responses to harmless molecules in the environment, things like foods or pollens, and can cause an allergy or hypersensitivity, or to self molecules, which would lead to autoimmune disease. The impaired immune system might fail to respond to pathogens or fail to maintain homeostasis with harmless microbes, which can lead to severe disease or immunodeficiency. These are various immune disorders that will be discussed throughout the upcoming topics. The immune system has 3 layers of defense. Each of the layers has its own specialized cells and molecules. There is the physical and chemical barriers, the innate immune system, and the adaptive immune system. The first layer of immune defense that is encountered by a pathogen comprises the physical and the chemical barriers of the skin and mucosal surfaces. Skin covers the outside of the human body, with an outer layer of dead cells and underlying layers of densely packed living cells, known as the epidermis and the dermis. These provide a strong physical barrier against the penetration by any pathogenic microorganisms. The regular shedding of the outer dead cells contributes to the physical removal of microbes. The skin does produce chemical defenses against invasion and colonization through the secretion of sebum, which has a low pH, is oily and waxy, and through sweat, which is high in salt content, by the sebaceous glands and sweat glands in the dermal layer. The mucosal surfaces line the cavities of the body that are exposed to air and ingested substances. The outer layer of the mucosa is alive, and it contains specialized cells that produce mucus, a thick and viscous fluid that coats the outer layer of cells, which functions to trap and expel any microbes. Cells that line the respiratory tract have short hair-like projections on their surface known as cilia. Failure of the mucociliary transport system can lead to lung disease, like chronic obstructive pulmonary disease, pneumonia, and an increased risk of infection. The antimicrobial enzyme lysozyme is abundant in mucus and tears. This acts as a chemical barrier for mucosal surfaces. Lysozyme functions by disrupting the cell walls of certain bacteria, causing the bacteria cells to rupture and die. The second layer of immune defense is the innate immune defense system that comprises fast-acting cells and molecules that recognize common features of pathogens. The broad response is orchestrated by circulating white blood cells and tissue scavenger cells. The innate immune cells become activated to ingest and destroy any invading pathogens. Cells of the innate immune response are generated continuously from bone marrow stem cells. Molecules of innate immunity include the proteins of complement cascade, a system that directly attacks and lyses bacteria. The cells of the innate immune system are able to interact and communicate with cells of the third layer of the immune defense, which is the adaptive immune response. The very specialized organs and tissues of the immune system are known as lymphoid organs and lymphoid tissues. These are separated into both primary and secondary based on their function. Primary lymphoid organs and tissues are the site of immune cell development. These will include the thymus, where the T cells develop, and the bone marrow, where B cells develop. The secondary lymphoid organs and tissues are the sites of adaptive immune cell activation, and these include the lymph nodes and lymphatic vessels, where cells respond to damage and infection of the tissues, as well as the spleen, where the immune responses to the blood-borne pathogens are initiated. Hematopoiesis occurs in the bone marrow, where precursor stem cells are supported to survive, proliferate, and differentiate by mesenchymal stem cells. The myeloid lineage can differentiate into granulocytes. These are the neutrophils, eosinophils, basophils, and mast cells. It can also differentiate into mononuclear cells, which includes monocytes, macrophages, and some dendritic cells. The lymphoid lineage comprises the T and the B lymphocytes, the natural killer cells, the innate lymphoid cells, and some dendritic cells. Here is the myeloid stem cell and its differentiation into mast cells, erythrocytes, basocells, neutrophils, eosinophils, and certain dendritic cells. This is the lymphoid stem cell and its differentiation into T lymphocytes, B lymphocytes, natural killer cells, and dendritic cells. Slower to respond than the innate immune system is the adaptive immune response that is highly specific for a particular pathogen and improves during the course of the response. T helper lymphocytes are modulatory and function to promote the activity of other cells. The B lymphocytes and the cytotoxic T lymphocytes are effector cells that are directly involved in targeting and destroying antigens and foreign cells. These are memory cells that are able to efficiently promote pathogen neutralization or elimination so that there won't be any clinical signs of infection. This is known as immunological memory. Immunological memory is the mechanism upon which a vaccine is based. The B and the T cells only recognize one type of antigen, meaning that each one has a single specificity. Here, the B cells recognize the antigen through their B cell receptors. The receptor has the same structure and specificity as the antibody that it goes on to secrete during an immune response. There are dozens of different options for each gene segment to combine together in a mix-match fashion to create these receptors diversity with very few genes present. This process is known as somatic gene rearrangement that occurs in the B cell precursors in the bone marrow. The B cell receptors and the antibodies are composed of 2 heavy chains and 2 light chains that form a Y shape. The stem is a constant fragment region that consists only of a heavy chain and has consistent amino acid composition that is determined by the antibody's class. The 2 antigen binding regions are made of terminal ends of the heavy and the light chains. The genes coding for these regions have multiple diverse segments that are shuffled during B cell development. Unused portions of the genes are discarded, and the remaining portions are rejoined in a relatively random fashion that produces the rearranged DNA. Because of the gene rearrangement, the proteins translated from the final light and heavy chain genes are very diverse. Each B cell recognizes its specific antigen out of millions of possible other antigens. And the millions of B cells constitute a repertoire that is able to respond to most possible antigens that will be encountered through one's lifetime. The thymus is a primary lymphoid organ, and it is critical for T cell development in early life. After 1 year of age, the thymus begins to shrink and its function is reduced. Thymocytes in the thymus undergo a process of somatic gene rearrangement that leads to the development of antigen-specific T cell receptors, similar to the B cells. Most of the T cells will express an alpha-beta T cell receptor, which is made of the alpha and the beta chains. The process of the gene rearrangement with variable and constant regions is similar between both the B cells and the T cells. Most of the T cell receptors are of the alpha-beta type, where the gene coding for alpha chains has several variables and joining regions and a constant region. And the gene coding for the beta chains has a V and a J regions, as well as diversity regions. Similar to B cells, rearrangement of coding sequences for both the alpha and beta chains creates a diversity of antigen recognition sites of T cell receptors and a broad repository of recognition sites for antigens that could be encountered throughout one's lifetime. Lymph flow is powered by muscle contractions during body movement, and backflow is prevented by one-way valves in the lymphatic vessels. The lack of movement or damage to the lymphatic vessels or valves can lead to an accumulation of lymph in the tissues, which is known as edema. This is often a common complication following radiation therapy or cancer surgery, and it is due to the damage of the local lymphatic network. The T and the B cells move into separate parts of the lymph node. The lymphoid follicles are predominantly populated with B cells and respond and proliferate in response to an antigen. In the spleen is where the T cells are mainly located, near the arteriole and in the periarteriolar lymphoid sheath, and the B cells are within the lymphoid follicle, which is adjacent to the T cells. Lymph nodes are located along lymphatic vessel pathways from the tissues to the thoracic and right lymphatic ducts that empty into the systemic venous circulation. Within lymphoid follicles, T cells and B cells that are specific to the same antigen can be drawn together and provide co-stimulation that promotes the development of mature lymphocytes, as well as antibody production and memory cells. The immune system comprises body mechanisms that protect from microbial invaders and cancerous cells at both the physical level, using rapid, activated, non-selective innate immune system, and a highly specific adaptive immune system. The innate immune response recognizes general molecular patterns of pathogens and rapidly recruits many phagocytic cells to both engulf and destroy pathogens. The adaptive immune response recognizes specific patterns of pathogens known as antigens, and this stimulates limited numbers of both T and B lymphocytes to respond to those antigens that will proliferate and generate the immune defenses. The innate immunity immediately leaps into action when protective barriers like the skin and mucosal membranes are breached. Another major function of the innate immune response is to vacuum up debris and pathogens during phagocytosis. Neutrophils, macrophages, and dendritic cells are highly phagocytic cells that will help to engulf and destroy pathogens. Tissue damage activates resident cells. Trauma damages tissue and allows pathogen invasion. This is signaled by danger and pathogen-associated molecular patterns. Mast cells and macrophages will release histamine and cytokines. Vasodilation and increased vascular permeability begin, thus increasing blood flow and the delivery of cells and proteins to the injured region. The white blood cells move to the injured area by chemotaxis. Cytokines, chemokines, and complement protein fragments will promote chemotaxis of neutrophils and monocytes to the injured region. Antibodies and complement proteins coat bacteria to aid in phagocytosis. Increased capillary filtration promotes local swelling and edema formation. Neutrophils phagocytose bacteria, killing them via an oxidative burst that generates reactive oxygen and fuse with lysosomes containing degradative enzymes, especially proteases. During inflammation cessation and repair initiation, the macrophages engulf apoptic neutrophils, cleaning up the injury debris while releasing anti-inflammatory mediators and cytokines to recruit fibroblasts and cells that are capable of rebuilding the tissue. In resolution and recovery, the macrophage signals stimulate fibroblasts and endothelial cells to support regrowth of the tissue and its vascular supply, therefore restoring tissue integrity. Neutrophils are highly motile, phagocytic, and become activated quickly in response to tissue injury and infection. Neutrophils represent 50 to 70% of all leukocytes. The immature cells, known as band or stab cells, are easily recognizable by the characteristic shape of their nucleus. Immature neutrophils are less segmented, appearing as a single band across the cell. Neutrophils are highly phagocytic. They are able to ingest and kill thousands of bacteria before dying. Neutrophils have two mechanisms of bacterial activity: exposure of bacteria to toxic oxygen compounds known as reactive oxygen species, and degradation of bacteria by protease and other enzymes. This is a neutrophil extracellular trap. In A, when the neutrophils encounter large numbers of pathogens, they can respond by extruding their DNA and granules in a sticky net that traps and rapidly kills many bacteria at one time. Here, this depicts an electron micrograph of bacteria that is orange, caught in neutrophil extracellular trap that is yellow. Although effective at the microbial level, because this is an extracellular reaction, it does have potential to create secondary tissue injury. Basophils and mast cells are related non-phagocytic granulocytes. These contain granules of pro-inflammatory chemicals such as histine that immediately after tissue injury or during the early phase of an allergic response is when these cells are deregulated, release their histine, which is a mediator of vasodilation. Eosinophils have a role in allergy and hypersensitivity reactions because they will often participate in a late phase of the allergic response. In tissue, monocytes increase in size and phagocytic capacity. When either infection or injury occurs, sentinel macrophages detect cell damage and migrate to that area by chemotaxis. Chemotactic substances that attract monocytes and macrophages include microbial products, components of damaged cells, chemicals released by other white blood cells, and peptides. The dendrite or dendritic cell is the most critical type for the communication between the innate immune system and the adaptive immune system. The immature dendritic cells are extremely phagocytic. A new group of innate cells that come from the lymphoid lineage are known as ILCs. These specific cells do not express the lineage markers of other immune cells, but they do respond to their local environment by recognizing cytokines, chemokines, and other chemical signals. The natural killer cells can recognize and directly kill cells that appear abnormal, like a virus-infected or tumor cells. These NK cells will scan for the presence of unusual ligands on cell plasma membranes. Once they are activated, these NK cells orientate and release their granules into the extracellular space toward the target cells. The granules contain mainly two substances, perforin and granzyme B. Perforin will create a channel in the plasma membrane. The membrane pores allow the entrance of the secondary compound, the granzyme B, which is a protease, to engage in the apoptosis, leading to cellular destruction. The toll-like receptors, or TLRs, are a subset of a pattern recognition of receptors that mediate pathogen recognition in the innate immune system. Many of these recognize components of bacterial, yeast, or parasite cell membranes. Others are found on endosomes and recognize components of invading bacteria and viruses. This is a non-selective method of distinguishing between self and non-self characteristics of the innate immune system. The complement pathway is a series of proteins that are produced by the liver and that can be found throughout the body in extracellular fluids, lymph, and blood. When it is activated by signals of invasion, the complement proteins initiate a cascade of activation where one enzyme cleaves and activates the next in the series. In the classical pathway, the bacteria are coded by antibodies that attract and activate the complement cascade. In the alternative pathway, the complement binds to the bacterial wall and it initiates its own cascade. In the lectin pathway, it depends on MLB, which is a circulating protein that is synthesized by the liver. So for it to attach to the bacteria before activating the complement cascade. The complement cascade is the target of regulatory proteins that suppress activation in the absence of pathogen invasion. Activated complement proteins are quickly degraded to prevent excess tissue damage. Mutations in complement genes may predispose the presence of a greater incidence of an infection. Cancer, autoimmunity, allergy, obesity, and metabolic dysfunction, as well as infection, genetic diseases, and responses to microflora and aging may all lead to chronic inflammation. So chronic inflammation occurs when an inflammatory response fails to resolve. This leads to increased and sustained inflammation that can last for months or years. Chronic inflammation will heighten the risk of developing many harmful diseases, including the cardiovascular disease, chronic kidney disease, cancer, and depression. The immune system comprises body mechanisms that protect from invaders and cancerous cells at the levels of physical barriers, which is the skin and mucous membranes, a rapidly activated non-selective innate immune system, and a slowly developing but highly specific adaptive immune system. The innate immune response recognizes general molecular patterns of pathogens and rapidly recruits phagocytic cells to engulf and destroy pathogens. The adaptive immune response recognizes specific patterns of pathogens known as antigens, and this stimulates limited numbers of B and T lymphocytes in response to those antigens that will proliferate and generate immune defenses. The presentation of exogenous extracellular antigens on MHC2 proteins is conducted by professional antigen-presenting cells. MHC2 presentation is a multi-step process that is initiated by the synthesis of MHC2. The antigen binding site of MHC2 is protected by an invariant peptide. MHC2 can move to the membrane or to an intracellular component, the MIIC. Foreign proteins are brought into the specialized antigen-presenting cells by endocytosis, forming the early endosome where they are broken down by proteases into smaller fragments. Fusion of the endosome with the MIIC allows the cleavage of the protein and replacement of the resulting clip with the antigen fragment. The final complexion of the antigen fragment and MHC2 protein are displayed on the membrane of these antigen-presenting cells for presentation to the CD4 T cells. In certain phagocytic cells, some of the proteins from the pathogen are broken down further in the phagolysosome into small peptides. These peptides associate with a transmembrane protein called the MHC class II or the human leukocyte antigen. The MHC1 is expressed by nearly all the nucleated cells. The CD4 T cells recognize the antigen on the MHC2. The CD8 T cells recognize the antigen on the MHC1. Intracellular antigenic peptides are presented on MHC1 molecules to the CD8 T cells, forming antigen degradation by the proteasome. The peptides generated are translocated via the TAP into the lumen of the reticulum. It's then loaded onto the MHC1 molecules. The peptide MHC1 complexes are transported to the plasma membrane by the Golgi apparatus for presentation to the CD8 T cells. The adaptive immune cells can recognize a specific strain of a pathogen, yet fail to recognize a different strain of the same pathogen. A remarkable feature of the adaptive immune response is the ability to specifically recognize and respond more quickly and more effectively to a second exposure with the same pathogen. This is the underlying principle of vaccination. The main effector mechanism of the B cell response is the secretion of antibodies. This is a specialized class of circulating glycoproteins that are a soluble version of the B cell receptor. Circulating antibodies are effector proteins that bind antigen on pathogens or toxins with high specificity. B cells are activated when the B cell receptors on their cell surface bind to free antigen. B cells often require help from T cells in order to be fully activated. In neutralization, antitoxin antibodies will bind toxin molecules into an antigen-antibody complexes that can be removed by phagocytes. In opsonization, antibodies bind to pathogens as osponins, facilitating macrophage phagocytosis. Antibodies binding to pathogens will initiate the complement via the classical pathway of complement activation, leading to MAC formation and cytolysis. Antibodies are made of 4 protein chains, 2 heavy and 2 light chains. These are held together by disulfide bonds and form a Y-shaped molecule. Functionally, these antibodies have 2 regions, one end with 2 arms to bind the antigen, and one end that is constant across their class and able to bind to receptors and recognition sites. The Fc region of the antibody interacts with different immune effector cells, like neutrophils, mast cells, killer cells, and macrophages. These bind to immunoglobulin class-specific Fc receptors expressed on the cell membrane. The 5 immunoglobulin classes or antibody isotypes are IgM, IgD, IgG, IgA, and IgE. Monomeric IgM and IgD are the first immunoglobulin types expressed on the surface of mature, naive B cells. These anti-surface antibodies function as the B cell receptor. IgM has the highest ability to activate complement to help destroy the invading pathogen. IgD is used as a marker for B cell maturation because its surface expression changes accordingly to different B cell maturation stages. IgG is the most abundant immunoglobulin and good at activating the complement cascade and eliminating pathogens. It also promotes opsonization and MAC complex formation on microsurfaces. IgG is the only isotope able to cross the placenta, providing protection to the fetus during pregnancy and the neonate in weeks after birth. IgA is secreted either as a monomer or a dimer, and it has the ability to cross epithelial membranes and to be expressed abundantly in body secretions that protect mucosal tissues and body openings. IgE binds to mast cells and basophils, interacting with its specific Fc receptor that will trigger the release of preformed granules that contain histamine and inflammatory molecules. During the first presentation of an antigen, either due to an infection or a vaccination, a delayed and relatively weak response will be produced, with IgM produced first and then transitioning to IgG. This stage results in B cells diverging to become either plasma cells or memory cells. The second exposure, or a booster shot, is when antibodies are rapidly produced, the total amount of antibody released increases, and there is very little IgM response. The clone of memory cells expands further. Further exposures to the antigen will elicit a robust response, providing protection that can last a lifetime. Concentrations of immunoglobulins, or titers, are specific for antigens on pathogens like rubella, hepatitis B, and measles can be measured in the blood. Analysis of antibodies for a pathogen-specific antigen can reveal whether an individual has been previously exposed to or vaccinated against that pathogen. And it can determine whether the infection is recent, as in IgM dominated, or has occurred in the past, then it would be IgG dominated. The T cell response, sometimes known as the cell-mediated immune response, involves the activation of T cells through the highly specific T cell receptors that recognize an antigen that's presented. The CD4 cells function to produce cytokine and are sometimes termed as T helper cells, where the CD8 T cells have cytolytic functions in addition to cytokine producing abilities. CD8 T cells are often referred to as CDLs, which play a critical role in combating intracellular bacteria and viral infections, as well as cancer cells. The antigen presentation to activate T cells, in addition to the T cell receptor, the MHC interaction, the effective antigen presentation requires additional co-stimulatory signals and changes in gene expression that is mediated by the transcription factor CREB in the T cell nucleus. The naive T helper cells have been characterized as developing into 5 different T helper subsets known as TH1, TH2, TH17, TFH, and TREG. The TH1 cells promote mechanisms of innate cells like phagocytosis and antigen presentation. The TH2 will support the B cell responses and drives isotype class switching to IgE, which is important for deregulation of mast cells and basophils to help fight helminth infections. The THFH, this supports the maturation and the proliferation of B cells. The TH17 cells can drive inflammatory disorders like autoimmune disease, multiple sclerosis. However, the TH17 cells are also necessary for maintaining homeostasis and barrier protection within the gut. The TREGs function to damp down immune responses, and these are essential for preventing autoimmune responses, providing tolerance to the gut microbia, and quenching inflammation once a pathogen has been eliminated. Mechanisms of cytolysis by CD8 T cells. When recognizing a virus-infected self cell, the T cell will release perforin, which penetrates the membrane of the infected cell, and granzyme, an enzyme that enters the cell and initiates apoptosis. In addition, the T cell FASL or FASL engages the target cell FAS protein, initiating apoptosis of the affected cell. A minor subset of T cells that also develop in the thymus are termed Y8 T cells. These cells express a TCR known as the Y8 TCR. Common in epithelial tissues like the skin and intestine, these cells are important for tissue homeostasis, thermogenesis in adipose tissue, and epithelial repair. These also contribute to immune responses against infection and cancer. Vaccination is a medical procedure in which immunological memory is induced into an individual without requiring an active infection. If sufficient individuals in a community are immunized against a pathogen, it is called herd immunity, and it will prevent disease transmission within the population. Vaccines routinely contain non-infectious material from a pathogen that still includes the pathogen-specific antigens necessary to stimulate the adaptive immune response. Live vaccines typically will elicit a more robust immune response and greater protection. Non-live vaccines will require the use of adjuvants, which will potentiate the immune response by activating the PRR receptors to drive an innate immune response alongside the adaptive immune response. Non-live vaccines will require more boosts than a live vaccine. Boosts occurring when an individual is revaccinated to create a larger antigen-specific memory B and T cell pool. The immune system mistakenly will generate an immune response to a harmless molecule from the environment, like food or pollen. Re-exposure to that molecule can lead to a strong inflammatory response that can cause varying degrees of discomfort and, in severe cases, even death. This type of reaction is known as hypersensitivity, atopy, or allergy. The response is mediated by the production of IgE that recognizes an environmental allergen. Upon later exposures, the allergen binds to IgE, causing mast cell and basal cell degranulation with the immediate release of histamine and the development of allergic inflammation. There is a critical step in the production of allergies known as class switching. This stimulates the B cells to change from IgM or IgG to IgE production. Because mast cell membranes have IgE receptors, the IgE molecules can bind onto mast cells and sensitize them to degranulate, which is the release of granules of histamine upon the next antigen exposure. The reactions that develop from type 1 hypersensitivity will include hives, eczema, allergic rhinoconjunctivitis, which includes hay fever and allergic rhinitis, allergic asthma, food allergies, and anaphylaxis. The type 2 antibody-mediated hypersensitivity occurs when a host cell surface molecule becomes modified by exposure to a chemically reactive molecule. This leads the immune system to perceive the host molecule as non-self. In the type 3 immune complex mediated, the IgM or IgG is produced against a soluble foreign protein. This leads to immune complex formation. Immune complexes are complexes of an antigen, usually with IgG, that circulate in the blood and can also form deposits in tissues and activate the complement cascade, thus leading to inflammation within that tissue and tissue damage. The type 4 cell-mediated hypersensitivity is known as delayed type hypersensitivity because it can occur several days after an exposure to an antigen. This will often manifest as a blistering skin rash. Autoimmune diseases occur as an aberrant and destructive immune response against self. In primary lymphoid tissues, which is the bone marrow and the thymus, only the B and T cells that do not respond to self proteins and therefore are tolerant to self are able to survive and mature. This is known as central immunological tolerance. Peripheral mechanisms of tolerance include inducing unresponsiveness or death in self-activated cells when they encounter antigen without any additional activating signals, and inducing self-reactive cells to become tregs or to be suppressed by the tregs. When both central and peripheral tolerance fail, autoimmunity can occur. Here are the general mechanisms in autoimmunity. The mechanisms underlying predisposition to autoimmune diseases aren't really known, but family history is routinely positive for members with one or more types of an autoimmune disease. It is believed that environmental exposures or prior infections can trigger autoreactivity. Autoantibodies are often present before any overt disease symptoms. Over a period of time, the autoimmune response will increase with tissue damage resulting from immune and complement activation. There are various biologic therapies that are being used for autoimmune disease. They are used in disease processes like Crohn's, colitis, RA, and asthma. There are two main categories of immunodeficiency. The first is primary. This is inherited or genetic immune deficiencies. And the second is acquired during the lifetime as a result of an infection, other diseases, a medical intervention, or environmental factors that occur like stress and poor nutrition. Primary immune defenses typically will present very early in life, as soon as protection against infection mediated by maternal antibody in the newborn has declined. For severe primary immunodeficiency, the best outcomes occur with hematopoietic stem cell transplantation. For specific immune deficiencies, regular IV immunoglobulin, careful monitoring, prophylactic antimicrobial use, and increased hygiene can help to prevent severe infection. The most prevalent cause of secondary immunodeficiency is HIV infection. HIV will infect CD4 T cells and some macrophages, and when left untreated will lead to a significant loss of immune cells throughout the body. This will result in a broad immune suppression known as AIDS. The immune system is significantly weaker in infancy and older adulthood than during any other time in life. At birth, a neonate immediately begins to experience colonization with commensal bacteria from the vagina during a vaginal delivery and from the skin in a cesarean delivery, as well as on the skin, intestinal, and respiratory tracts thereafter. In older adults, the Th2 responses are preserved, while activity of Th1 and B cells declines. Infections like influenza have a higher morbidity, mortality, as well as a risk of developing secondary bacterial pneumonia. The trajectory of colonization with normal flora and infant microbial diversity is influenced by the mode of birth, whether it's vaginal or cesarean delivery, breastfeeding versus bottle feeding, antibiotic exposure, and introduction introduction of solid foods, as well as exposure to various environmental conditions. Microbial diversity influences immune development and may influence later a risk of autoimmune and hypersensitivity disorders. Atopic dermatitis. The immune system activity decreases with age, with adaptive immunity impaired more than the innate immunity. This is referred to as immunosenescence. There are two opposing changes in innate immunity. One is a decreased function of neutrophils, macrophages, monocytes, dendritic cells, and natural killer cells, which increases the vulnerability to infections. While, secondly, there is a general increase in the inflammatory mediators. The concept of inflammaging is based on observations that the levels of inflammatory mediators generally increase with aging.